All right, thank you, Paul. It's an honor to be here and to share with you some of the work that we're doing in my group and in this field. So I'm sort of representing the biological perspective here. You'll see, though, that there really are a number of points of contact with probably all of the previous speakers to some extent, and certainly with Sharon's talk in terms of entropy as a driving force potentially for self-assembly in cells. So if we think about biological systems, there's an enormous degree of richness to the sets of interactions. There are thousands of different types of molecules, biomolecules like protein and RNA and DNA that are interacting in a crowded environment. And so the interactions look much like you see in the simulation on the left. Those interactions, despite the enormous number of fluctuations, thermal and likely non-equilibrium fluctuations within the system, those interactions give rise to coherent assemblies, so coherent organization. Things like the migrating cell on the top, the leading edge of the migrating cell, or this neutrophil chasing a bacterium in the middle, or a frog oocyte or embryo that's undergoing gastrulation in the bottom. So I think it's fair to say that biological systems are the most richly textured form of matter in the whole universe. And you know, if anyone has something more richly textured, I guess we'd have to define that, but then let me know. But I suspect we're going to have to wait until we find an alien life form, and maybe it'll be more highly evolved or something. But that's a call to arms. I mean, in terms of physics, we suspect that there's going to be entirely new physics in biological systems by trying to study the principles that govern self-assembly. So what makes biological systems so interesting, one of the features that makes them so interesting is that there is a wide span of length scales involved and sort of hierarchical self-assembly. So you can think about, at the nanoscale, the building blocks, protein and RNA interactions, let's say, and somehow those interactions encode for organization within the cell, things like organelles, and then the function, the assembly and function of those things then dictate cell behaviors, tissue behaviors, and organism behaviors that ultimately can build soft, squishy robots like you and I. Now, what's interesting, I think, about this is that we actually know a fair amount, and certainly not everything, but we know a fair amount about the physics that governs self-assembly at the nanometer scale, so at this kind of smallest scale, as I represent here, of biological self-assembly. We can think about structural biology models, protein folding models. We have a pretty good conceptual framework for understanding things at that level. If we go up to the highest length scale, sort of what I'll call sort of macroscopic length scale, we can think about continuum mechanics models, the kinds of cell vertex models that people think about in collective tissue behavior, or even, if you want to stretch, so you could say, sort of physics-inspired behavioral models. So we can say something about what's happening at that scale. The length scale that I want to talk about and is the focus of the discussion today is this mesoscale at the sort of micron length scale, structures within living cells. And I would argue that we have very little understanding of the rules that govern assembly at that scale. So I'm going to try to tell you about some of the ways we're thinking about that. A lot of the work in my lab and in this field that I'm going to tell you about is inspired by soft condensed matter systems that are non-living but have some features, qualitatively at least, that are similar to biological systems. They're squishy and they involve macromolecules, things like polymers. They can give rise to emergent behaviors, viscoelasticity, and so forth. So we're thinking a lot about the ways in what we've learned in soft non-living matter and how we can take some of those insights into living cells and ask, can this give us new insights and new knowledge on how cells are organizing their contents? So if you think about what's happening inside living cells, that picture, the movie I showed you in the beginning, the simulation of all these molecules that are interacting. One of the ways, sort of high school biology textbook picture of how the cell organizes its contents is through organelles. And so many of you probably remember from high school biology the organelles with these funny names like endoplasmic reticulum, Golgi apparatus, and so forth. So essentially all the ones we learn about in high school biology are membrane bound, and they're to some extent vesicle-like. They're like little soap bubbles that compartmentalize the inside of that, of that compartment from the outside. And so that kind of makes a lot of sense. But we think that that's probably just really the tip of the iceberg in terms of the way compartments form inside the cell and the way in which the cell organizes its contents. Turns out there's a whole broad class of membraneless organelles, so bodies that are large-scale assemblies of thousands or millions of molecules that are forming coherent structures inside cells that play a broad range of biological roles. I'll tell you a little bit about these germline P granules in the top left here. There are things like processing bodies, signaling assemblies, and there's an array of these structures inside the nucleus as subcompartments of the nucleus. And in fact, I think it's probably fair to say that most of the bodies inside of cells, most of these kind of organelles that one sees are in this category of membraneless. So it's not, it's not sort of a small subgroup, but in fact, maybe most of the organization is mediated by these kinds of structures. So I started thinking about these in the context of this problem, which these structures are called P granules, and they're implicated in specifying the lineage of the cell, so the fate of the cell, whether the cell is going to form a neuron or a gonad or something else. And so these structures are implicated in specifying the reproductive organ, so the gonad of the C. elegans worm. And what's interesting is initially, after fertilization, you can see that they're distributed throughout the embryo, so these green puncta distributed throughout the embryo. But as the embryo starts to polarize and develops a distinct anterior versus posterior axis, these structures end up on one side. And when the embryo then divides in two, only one cell, the cell on the right, gets those structures. And that's the progenitor germ cell that goes on to make the reproductive organs in the worm. And so this process, we've learned a lot about how it works. It's mediated by gradients of molecules that are kind of morphogens that control the segregation of P granules into the embryo posterior. What's really fascinating about how this works is that these structures, these P granules we discovered are liquid-like assemblies of RNA and protein. So despite the name as a granule, they're not little pebbles. They're actually condensed liquids. And you can see that in this movie where we apply shear stresses across a tissue in a very kind of low-budget rheology experiment. We just basically squash this tissue and induce shear stresses. And what you can see is these P granules flow and drip and wet and coalesce with one another. And so this led to a model wherein the segregation of these P granules to the posterior end of the embryo is a kind of phase transition or a liquid-liquid phase separation process, just like what Sharon Glotzer was telling us about in a non-biological context. So the idea is that the anterior-posterior axis of this embryo spans the two-phase boundary such that the posterior end of the embryo is in a two-phase region where these condensed liquid states of RNA and protein are stable. And since we first put forth this model, there's a lot of data supporting this, and it's a very rich problem, and there are many non-equilibrium features which I want to highlight. The cell is very much out of equilibrium. You can see already from the gradient that drives this assembly. So this idea of condensed liquid states inside the cytoplasm and phase transitions driving organization inside cells actually has a long history. We started to discover papers back in the literature as far back as 1899. Pioneering interdisciplinary biologist E.B. Wilson had some of these ideas about the physical nature of the cytoplasm, and he described the cytoplasm or the protoplasm as a liquid in the form of an emulsion consisting of these suspended droplets. In fact, we've known that biomolecules can undergo phase transitions for many decades, and there's a lot of work with structural biology where trying to induce proteins, for example, to crystallize such that you can do X-ray scattering. Phase transitions have also been suggested to potentially play a role in membrane organization. And so there's kind of hints of this in the literature. And it has started to become apparent that these kind of phase transitions are ubiquitous within the cytoplasm and the nucleoplasm, but the contents of the cell. There's literally dozens of different, or really at this stage, hundreds of papers over the last several years that have come out suggesting that liquid-liquid phase separation is implicated in processes, ranging from mitosis to cell signaling, postsynaptic densities that mediate connectivity in neurons, and essentially, without exaggeration, I would say almost every biological process that's happening in a cell, there's some connection to liquid-liquid phase separation now that has been described. So we're really interested in this process, phase separation, and how it is occurring potentially to organize the contents of the nucleus. So the nucleus is where the genome sits. And so we're all familiar from 23andMe or something, with the idea that our genes are dictating phenotype. What's something that you may not appreciate is that every one of your cells, so this is actually a picture of my daughter when she was first born, every one of her cells has the same genetic code, the same sequence of base pairs that are supposed to dictate the phenotype. And yet she has dozens or hundreds of different types of cells that make up her body. So each one of those cells, its phenotype is supposed to be dictated by the genome, and yet you get totally different cell types with different shapes and different functions. So how does that work? Well, it turns out that it's becoming apparent that these kinds of condensed liquid states are like little dewdrops that are sitting on the grassy genomic landscape, and they're playing roles in mediating expression of genes. And we've started to refer to this as a liquid epigenome. So there are many different types of these condensed liquid states which we think are doing this inside of cells. Structures like the nucleolus, which I'll tell you about, which controls ribosomal RNA biogenesis, so the protein translational machinery inside of a cell. Structures like nuclear speckles, which are regulating splicing, so how genes are being cut and cutting out introns, and so forth. And Cajal bodies, which are also playing roles in the regulation of expression of genes. So we started studying this with a postdoc in my lab, Steph Weber, who's now a faculty member at McGill University. And she was able to show that the nucleolus, so this structure that's inside the nucleus, the nomenclature is a bit unfortunate. It's the nucleolus, and it assembles around these particular genomic loci that make the ribosomes. And she was able to show some evidence that the assembly of this structure seems to follow a phase boundary. This is we could change the concentration, and the higher the concentration, the more material condensed out of the nucleoplasm. We could also look at the dynamics of the assembly of these droplets and show that there was coarsening of nucleoli around those transcriptional sites that was consistent with sort of classic modeling approaches. So this is the Cahn-Hilliard equation, which is a kind of diffusion equation, except it's uphill diffusion. I was going to make a point about how entropy can only drive things downhill in Fick's law, but that may be totally wrong now that after Sharon's talk, so I'll hold my tongue on that. But it's exciting to think about. But generally, one thinks about diffusion down concentration gradients driven by entropy. And so if you want something that's uphill diffusion, you would have to have some interactions, something beyond just entropy. And that's what's in that formalism. What's different here, though, is that we have the presence of RNA due to transcription at a particular genomic locus. And that's why the droplet in the middle wins out. Another system that we've been using to probe this idea that liquid-liquid phase separation is important in the nucleus, in particular with the structure of the nucleolus, is a frog oocyte system. It's really powerful because it's a single cell. It just grows to be very large. It grows to be about 1 millimeter in size. So it's a single cell that you can hold in your hand. And it contains a nucleus that is similarly large. You can also see it with the naked eye. And inside that nucleus, there are hundreds of these bodies, hundreds of these nucleoli that are sitting there and can be mechanically probed and tested in other ways. And so this is actually one of the first systems we started studying, and we showed, after the pea granule story, and we showed that these nucleoli really have classic liquid features. This is a liquid bridge rupture after they started to fuse, and we managed to pull them apart. What's an interesting feature here is, again, there's signatures of non-equilibrium, the importance of non-equilibrium in cells, because if we remove ATP, which is sort of the molecular battery inside the cell, then the apparent viscosity of these structures goes up a lot. So they tend to fuse much more slowly, and their dynamics are slowed. So that's something that we're kind of actively studying. First graduate student in my lab, Marina Farik, who's now a postdoc at the NIH, came at this problem and started thinking asked a very simple question, which I'm a big fan of asking very simple questions at the outset of a project. And so Marina asked the question, well, if these are all droplets, why aren't they all coalescing? What's keeping them from all forming one, mega droplet, like, in salad dressing if you shake up the bottle and then come back? And so what Marina found was that inside this nucleus, there's a scaffold which is formed by an actin network, which is very interesting because it's a nuclear actin network. We're used to thinking of actin in the cytoplasm. And that actin network supports these droplets and keeps them from coalescing together. And what Marina found is that if she disrupts the actin network, these droplets actually do all coalesce. What we expected was going to happen was that they would all diffuse around and bump into each other, but what we found is they actually sediment under gravity to the bottom of this nucleus. And that was pretty surprising to us, because I had actually been teaching in class how gravity is negligible in cells. And so Princeton didn't fire me, so that was good. But the calculation you want to do is you want to calculate this sedimentation length scale, which is essentially reflects the competition between entropy, which wants to mix up a system, so sort of captured by KT, and the gravitational force, mg, or here expressed in terms of a buoyant mass. And what's interesting here is that the cell has grown so large that it's actually exceeded this gravitational length scale. So that's like the equivalent of climbing a mountain in Bolivia and you can't breathe, right? The physics is the same. You've sort of crossed this length scale. And in fact, if you do this experiment, if you wait overnight, you do find one single large droplet. And so this is the world record nucleolus. It's about 100, it's bigger than most cells, so it's this giant thing. So the other thing that's is really interesting about this work is that Marina found when she was doing these experiments and inducing this coalescence by disrupting the scaffold, she found that there are subcompartments of the nucleolus that actually themselves also coalesce during this large-scale nuclear coalescence. And so I'll show you this movie. The green shows an interior compartment that's characterized by this protein fibrillary. And the red is this outer compartment characterized by a protein nucleophosmin. And what you can see is that as these nucleoli start to coalesce, the inner cores also coalesce. And one has a sense that what you're seeing here is a kind of liquid within a liquid, as you can see in these higher resolution images on the right. And in fact, that's exactly what we think is going on. We were able to show that we could reconstitute this liquid within a liquid assembly in vitro with purified components. What's remarkable about that in vitro reconstitution is that the core shell architecture is maintained. So these green proteins, fibrillary in particular, is on the inside and nucleophosmin is on the outside. And that, it's known from multi-phase liquid physics that the droplet with the higher surface tension with respect to the surrounding fluid will be engulfed by the other droplet to minimize that surface energy cost. So it's basically, if it's a high surface tension, surface energy, then if it's engulfed by the other droplet, it will, that interface will go away and the energy will go away. And so we were able to do some surface contact angle measurements and surface tension measurements to show that that was consistent. It was also consistent with computational work that we did together with Rohit Papu's group at Washington University in St. Louis. So we like thinking about this because there are dozens of different types of these structures, and often they do have subcompartments, or there seem to be multiple liquid-like phases, which includes things like the stress granules. So we're interested in this idea that the relative surface tensions again structures the contents of these other bodies. There are other there are other bodies that don't interact at all. So they're not engulfing one is not engulfing the other, but they're not fusing or coalescing or interacting at all. And so we think that reflects a high surface tension that would be associated with the interface between those two. Again, it's known that in multi-phase liquid systems, you would have complete exclusion of those droplets. There are other biological systems where there's partial engulfment of the droplets, one partially within the other, which is associated with sort of relatively more equal surface tensions. And things like this beautiful image of this Cajal body, and the attached structure is called the snurposome, which I always think is such a cute name, especially for something that looks like Mickey Mouse here. But this is a case we think of a partially engulfed liquid. So I haven't said much yet about the molecular driving forces that are at play here. It turns out that many of the proteins that are driving phase separation are referred to as intrinsically disordered proteins. So they're proteins that are conformationally heterogeneous. So I think everybody knows that proteins are linear polymers. They're linear chains of amino acids. But we generally don't think of them as polymers because we think of them as having a compact three-dimensional shape that dictates their function. But it turns out that many proteins are actually conformationally heterogeneous. So rather than being folded into a single compact shape, they're actually sort of fluctuating around. And I'll show in this movie, this is a simulation of a protein like this, one of these intrinsically disordered proteins, or IDRs. These are closely related, by the way, to what are referred to as low complexity sequences or prion-like domains. And so we and others in the field over the last few years have been intensely focused on these IDR proteins that contain IDRs, because when you purify these proteins and look at them in vitro, what you find is that they readily phase separate into these condensed liquid states, which we think correspond to the same kind of molecular driving forces as the corresponding structure in the living cell. So this includes proteins like LAF1, which is one of a set of important IDR-containing proteins in P granules, these germline specifying structures that I told you about at the very beginning, and also with nucleoli in the bottom two examples. And then this example, the yellow droplets, that's from a fungus. So these are just a few examples from our work, but there are really many dozens at this point of intrinsically disordered regions which are thought to drive phase separation. Another key molecular architecture of proteins that are driving phase separation is oligomerization. So a lot of the proteins, and we've just really started to fully appreciate this pretty recently, is that a lot of the proteins have they'll have these intrinsically disordered regions. So this is a linear representation of the domain structure of a protein. You'll see that blue intrinsically disordered region in the middle. On the left is what's commonly seen as there's an oligomerization domain. So that could be a dimerization domain, so homodimerization, or the protein I already discussed, nucleophosmin, it has a pentamerization domain. And so some sort of self-interaction that's can be of that flavor. And then they'll often have a substrate binding domain, which could be an RNA binding domain. And so RNA is also in the mix here in many cases, and DNA can be as well. And so we started thinking about about these architectures where you have oligomerization and intrinsically disordered regions, which are interacting with themselves, we think, in a sort of weak multivalent fashion to drive phase separation. The issue in the recent developments of this field is that there has been very limited tools to study phase separation, particularly in the context of a living cell, embracing the full complexity of the cell. And so my group has been investing in developing technologies to be able to probe and control phase separation in living cells. And this involves an effort from a number of individuals who are shown here. I want to highlight in particular Youngdae Shin, who's a former postdoc who's now a faculty member at Seoul National University, who really got this work started. It's been continued in next generation versions of this by a postdoc, Dan Braha, a graduate student, E.J. Chang, and we benefited enormously from collaborating with my colleague, Jared Tocher. The idea with these systems is we're taking advantage of optogenetic proteins. So these are proteins typically borrowed from plants that can interact in a light-dependent fashion. So you shine light on these proteins, and something changes about their conformation that can open up binding pockets or interaction domains. And so the idea was we would have a light-dependent oligomerization. And so I'm going to show you a little bit about some of these systems. The first generation was this optodroplet system, and then we have a correlate system and a cast drop system. I'll tell you about the latter two. So the correlate system is really powerful. What it is it's built around a self-assembled scaffold, a 20-former. We're primarily using this protein self-assembly ferritin. And we've engineered each of those subunits to have one of a light-dependent heterodimerization domain. The other, the light-dependent binding partner, the other of the heterodimerization domain, we hook up to whatever protein we want to recruit to that core. So the idea is we shine light on the system, and then we get this core that now is decorated with these interacting domains that typically have the intrinsically disordered regions. So I'm going to show you these cells that are expressing these constructs, and we shine light on them, and you see that you get phase separation within the nucleus of these cells, and forming these condensates within the nucleus. You can have a lot of fun with this. This is toggling the light in time and asking questions about reversibility. And of course, since it's light, we can shine the light wherever we want and make pretty pictures. This is a 3x3 array of droplets, and that's evil droplet man, as you can see. So we are having a lot of fun with this, and there's a lot of sort of non-equilibrium physics we're exploring. If you induce phase separation in one part of the cell, what happens to the other parts, and things like that. One of the things that I'm particularly excited about is, we've seen in the previous talks different kinds of phase diagrams, or discussion of the rich phase behavior, the different crystal lattices associated with different regions of the phase diagram, and so forth. This is a phase diagram of steel. So this is kind of, in some way, it's a cornerstone or bedrock of materials physics or condensed matter physics. We try to be quantitative about the interactions and the underlying and the phase behavior. In biology, we still don't really have anything like that. So this is a schematic of what we think might be happening, where you have molecules that condense into liquid state and then we think can solidify. But we really don't have anything like that. But these optogenetic systems have allowed us to start to map phase diagrams in cells for the first time. This is a phase diagram that has this kind of characteristic binodal inverse paraboloid shape that is commonly seen in polymeric systems. And so the solid red circles are cells at those concentrations and a oligomerization state that do phase separate. The open red circles are the ones that don't phase separate. And these other points, this is the concentration of the condensed phase and the concentration of the dilute phase. And so we can explore this. We can start to ask questions. Do we see nucleation and growth? And we do. We see things like spinodal decomposition, and we're starting to be able to define the spinodal. And we also see some evidence for gelation boundaries that cut through these binodals. And that's really interesting because many of these proteins that we're studying are also implicated in neurodegenerative diseases like ALS, so that's Stephen Hawking the disease, and Alzheimer's, for example. So in the remaining time, I just want to tell you about some of the ways we're using these kinds of tools to probe the contents of the nucleus. There's been intense interest in the last several years in what's happening with these structures in terms of regulating gene expression and the organization of the genome. But we really have no idea still what's going on. And a lot of that, I think, is because of this lack of tools. And so we feel that we're positioned to be able to answer some of these questions. What really are these droplets or these condensed liquid states doing in the nucleus? How are they interacting physically with the genome to regulate the expression of different genes, to control the three-dimensional organization of the genome, which dictates its expression and function? So to get at this, we developed this optogenetic system, which is called Cas-drop, and it's built around the CRISPR-Cas9 revolution that you may have heard about, that allows you to sort of direct things to individual genes. And what we've done is taken that Cas9 and hook up to it a light-dependent oligomerization platform that allows us to drive phase separation at a definable, programmable genomic locus. And so here these are telomeres labeled in white, and then I shine light on the system, and you can see we get phase separation at those defined genomic loci. And so we can study the nucleation and growth dynamics of this, and we get off-target droplets if we ramp up the saturation too quickly, and so forth. In doing these experiments, we noticed something really interesting, which we're still trying to wrap our heads around and understand. But in doing these experiments where we see some of the off-target droplets being nucleated in control experiments where we don't guide the Cas9 to a particular locus, what we noticed was that the droplets, as they grow, they tend to exclude the genome. They tend to exclude chromatin as they grow. And I'll show you movies of this. What you're going to see is some holes starting to appear, and those holes will appear exactly where the droplets grow. And so you can see that here. I could play this movie again since it's sort of fun to look at. But basically, we've got little black holes that are forming inside of the nucleus as these droplets exclude chromatin. And so what's been something that we're really interested in is what does that mean mechanically when you have coupling between phase separation and the mechanics of the genome? After all, we think of our DNA as software, but it's also hardware, right? It's a physical object with a persistence length and so forth. So it's a kind of entangled polymer network that has some viscoelasticity. And the elastic component of that network can actually contribute to the overall energetic calculation if we're thinking about whether these droplets will form, where they'll form, and so forth. We were inspired, I want to just highlight, we were inspired by this beautiful paper in a non-biological system by Eric Dufresne's group, and started to look for these effects in our system. And I'll show you that's what we think is happening. There's a mechanical effect. And the reason for that is, I should go back and just show you, the idea here is that if these droplets try to nucleate in particularly stiff regions of the genome, then they're going to have a hard time compared to the softer regions of the genome. And it's known that there's a lot of heterogeneity in the mechanics and the compaction state of the genome. So this is a case where we try to nucleate droplets within a dense, what's referred to as heterochromatic region of the genome, a transcriptionally silent region of the genome. And when we do that, so I'll try to put a droplet in this dense region of the genome. What you see is that these red droplets do not go inside that heterochromatin region. Instead, they decorate the outside of it like flower petals. And we think that that's a mechanical exclusion effect. We can actually simulate that together with our collaborators, Joel Berry and Miko Hataya. We can simulate that mechanical effect, and we think it reflects this mechanical energy deformation contribution to the phase behavior. So I'm just about out of time here, so I just want to make a couple last points. One is that these droplets can still pull genomic elements together through coalescence. And we can see that here, and we think we're able to use that to actually measure some of this the mechanical stiffness of the genome. What that means, though, is that the droplets, if they're attached to particular genes, they can pull them together through coalescence and actually activate genes that way. But non-specifically interacting parts of the genome are excluded through this exclusion effect that I showed. So we think this is a kind of way in which phase separation is rearranging the genome in a kind of smart way, filtering out the non-targeted regions of the genome and bringing together the targeted regions. So I'll try to tie this up here now. At the beginning of the talk, I told you that a key kind of poorly understood part or length scale of biological organization is this mesoscale level. We think that liquid-liquid phase separation is a key aspect of organization on that scale. We think many of these organelles, really dozens of different types of organelles in the cell, are assembling by liquid-liquid phase separation. There's a lot to figure out, and there's many ways in which we think there's going to be new physics at play that go far beyond the sort of equilibrium statistical mechanics pictures that we're using now, because that's kind of what we have. And I think that the multi-component non-equilibrium features, it's a whole playground for condensed matter physics, and I hope you'll join me in this adventure. So I think I mentioned key individuals as I went through the talk, so I won't list them again here, but I just want to thank our group. We have a really fantastic and interactive group, and I'd like to thank the people who funded us over the years, and thank you again for your attention.
Wonderful. We have some time for talks. Yes, please, over on the right.
Professor Brangwynne, thank you very much for certainly one of the most beautiful talks I've seen this week. Congratulations on some amazing discoveries. I had a couple of questions on some of the details of the phase separation. So you showed some pictures of spinodal and binodal decomposition, and then the more classic balancing the surface energy versus the bulk energy to drive the phase separation. And so in that movie that you had of the droplets that were trapped in the actin network, that essentially prevents the droplets from coalescing and growing that way. But you would also expect, if this were an equilibrium process, that you might see the analog of some Ostwald ripening or some smaller droplets disappearing at the expense of the bigger ones, and the actin network would prevent the coalescence, but you should still imagine some distribution maybe of smaller droplets disappearing. So is that a sensible way to look at it? And I didn't see, at least in the movies I saw, did you see small droplets disappearing while the larger ones grow in cases where coalescence is blocked?
Yeah, this is a good question. So Ostwald ripening effects, we think are playing a role, and so we explored some of that with the nucleolus in the experimental system of the C. elegans embryo. In the frog oocyte system, you don't usually see shrinking of those droplets, the small ones at the expense of the large ones. I mean, the Ostwald ripening effect, as you know, is ultimately mediated by surface tension, and these surface tensions, as for other macromolecular liquids, tend to be small. So you sort of order of magnitude say the surface tension would be kT over some molecular length scale squared, and these are macromolecules, so the surface tensions tend to be low. And so we think that Ostwald ripening is less important here than maybe in some other systems.
You wouldn't expect proteins to diffuse through these distances between droplets and coarsen that way.
Yeah, I guess what I'm saying is there are places where we think Ostwald ripening is at play, but in that frog oocyte system, we don't usually see that. And I think most of the droplets themselves are already quite large, and the surface tensions are small. So we think it's less, it's not. That's why it's not visible there.
Thank you.
Yeah.
Let me take a question from the left-hand side.
Hey, Cliff, it's Sarah.
Hey, Sarah.
Hi. I want to learn a little bit more about the nucleolus. Is it always the case that you have NPM1 and fibrillarin phase separating within the nucleolus, or is that only under particular conditions?
Yeah, so those are, that's generally, that's what you find in the textbooks, and that's what we see in these systems. I mean, they are in separate compartments. I mean, there's some partition coefficient, right? So you can have some NPM1 within the FC, within the fibrillarin rich, and vice versa. And we actually are actively studying, the partitioning and sort of the physics of partitioning, if you like. But yeah, that's not induced. There is a way to induce a sort of a higher order demixing if you turn off RNA transcription, which I could tell you about, offline, but that's the native state.
Okay, cool.
Yeah.
Good. I think I'll request further questions, hopefully right after this.
Sure,
I would
yeah.
like to thank the speaker and all the speakers.